So it's an honor today to present on behalf of the Carcinogenomics Project working team, alongside of Chris Corton as well. And if you go next slide. So this is just the disclaimer for Chris that the views he expressed do not reflect UPA policy or endorsement by the US EPA. So next slide. So before we begin, I just want to give a big thank you to this team. We have representation from 12 different industry partners, 9 academic and nonprofit partners, and then 6 government organizations as well. And I think you'll see is we've accomplished quite a bit over the last few years and this year as well. And so really big thank you to everyone who's contributed to the project. And like others have said too, we're always interested in new participation. So if there's interest or you just want to follow along, please reach out. Excuse me. So the goal of this project is to develop biomarkers that will help us detect tumor genesis sooner than we can see by gross histopathology, which generally requires a 2-year rat assay or rodent assay. And so we're taking a 2-pronged approach to this. One is to look for biomarkers indicative of induction of molecular events that are known to lead to tumor genesis in the rodent or humans. And typically those markers we're finding we can detect within days of treatment. So say in a 1-week study. And then we're also looking for technologies that can detect early clonal expansion and tumor genesis earlier than we could generally see by gross pathology. And so that's the second arm. And as we outlined in our Toxicology publication this year from the working group, we really see that the combination of these 2 approaches will help form a powerful way to evidence alternative that we can present aligned with ICS S1B, the revision that was just accepted for potential waivers of the 2-year CARSO studies. You can go next slide. So with that, the objective of the group then becomes to derive international industry, regulatory, and academic acceptance of this weight of evidence approach using these new genomic tools, using them in short-term in vivo rat studies to inform on the tumorigenic risk at the end of 2 years in the 2-year CARSO studies. Again, we have 2 aims. What we'll focus on today is biomarkers of the molecular initiating events or key events upstream of tumor genesis. And then tomorrow we'll be talking quite a bit about the application of error-corrected sequencing as a way of more early identification of mutagenesis or clonal expansion. So the next slide goes into more depth what we mean by these molecular initiating events or MIEs. And so we're fortunate that multiple AOPs are understood of mechanisms that can lead to tumor genesis in the rodent. We've so far focused on liver for 2 reasons. One, is the most common site of tumor genesis in response to chemical carcinogens. And then the second is that our liver happens to be where we have the bulk of the transcriptome data in order to derive our signatures. Our hypothesis is that sustained activation of these MIEs, and that could be DNA damage through a genotoxic mechanism or activation of non-genotoxic MIEs. So that includes nucleoreceptors like AHR, ER, CAR, PXR, PPAR, as well as just sustained cell proliferation that you get from regeneration from cytotoxicity. The hypothesis is that maintained activation of those mechanisms will lead to tumors at the end of 2 years, and measuring these mechanisms early on. And we know that each of these mechanisms is a potent inducer of gene expression changes in liver. So using those gene expression markers in short-term studies will help us predict the outcome at 2 years. We're not completely working in the dark here. There's been a number of papers over the past years that have shown the proof of concept of doing this. And so really the goal of the working group is to align on the identification of these gene sets, what the optimal genes are, and what the threshold should be for predicting carcinogenomic outcome. So this slide is an overview of how the group is organized. So there's 4 different subcommittees. There's a strategy and outreach leadership team that aligns on the overall vision and oversight, and also coordinates the outreach for the team. The heart of the group is the signature development team. This is the group that's going to be deriving the biomarkers and the thresholds that we'll be applying. But before we can do that, we need data. And so the data compilation team was tasked with bringing in all the genomics data that's available that we'll need to derive the signatures. And then we also need to classify our compounds by carcinogens, non-carcinogens, and which mechanisms they activate. And that's been the work of the mechanisms team there in green. So what I will do is just step through each of these then and give you an update on the progress that these teams have made this year. So here is the strategy and outreach team. We already mentioned multiple times today this paper that was published earlier in the year in Toxicological Sciences that really outlined our strategy and vision for the approach, and acts as a really nice communication to the world of the work we're embarking on. We also presented that as in poster form at SOT this year, and have submitted an abstract for next year to give an update on the progress. The data compilation team mostly completed last year, so it hasn't been a ton of work needed this year. But in the previous years, that team was able to compile 6 rat data sets. And if you jump quickly to the next slide, it outlines what those are. So there's 4 Affymetrix data sets, the drug matrix, TG gates, the iMarkar data set from Bayer that were compiled, as well as 2 RNA sequencing data sets. There's a data set from Merck of 95 compounds, and then SEQC also sequenced a portion of the drug matrix data set, so that's included as well. So all that together gives us data on rat liver data treated with 370 unique compounds, ranging from hours after the first dose to 30 days of dosing. With that in hand, then the data compilation team accessed all the original raw data for all those studies, brought that data in-house, normalized it, QC'd it, consistently ratioed to controls. And that's all been available on the SEBS server. And I'll show you the slide showing that in a second. But then we also used common nomenclature for all the chemicals, so it would help us align with the compound classification team, so we can align our annotation tables downstream. So we just jump 2 slides ahead then. This is an example of how the data is archived and available for use on SEBS. So all the QC reports are there, the raw cell files are there if folks want them, but then also the normalized data, the ratio data, and the outliers, et cetera, are all annotated. Okay, so the compound classification team has made tremendous progress, particularly this year with the use of, we reached out to a number of graduate students that helped us with this annotation. Turns out, it's a Herculean task to scour the literature for each of these compounds as far as the 2-year carso data, but then also the molecular initiating events that are associated with each of these compounds. And so it's been a ton of work, and a big thank you to this whole team, and especially the students that have helped really dive into the literature. So we have completed the annotation of all the compounds and the MIEs that they activate. And if you flip to the next slide, here's an example of just all the information the students were able to compile. You obviously can't read this slide, but for each of the compounds was annotated with the individual MIEs that we believe they induce. And then all the supporting data is then listed to the right-hand columns. So things that the students looked into are, gene expression data sets, chip data sets that show regulation of the transcription factor binding, mobility shift assays, knockout studies that show the requirement of the MIE for downstream activation, and then all the associated publications are captured as well. So really a big amount of work, but really nicely now provides us the data and the weight of evidence for each of these compounds and the MIEs that they activate. Go back one. Sorry, I'm jumping around. And then for the cancer annotation, we've looked into a number of different sources, including some of the already curated data from LASA, publications like Brambilla et al., the NTP, as well as going to the product labels as well. And the summer intern this summer was able to compile that information for 140 compounds. So about half the set. We still need to work through the rest, but we've captured most of the well-known ones that have Carso data. We're not going to have Carso data on all 370, but we still need to do go through the fine-tooth comb with the rest and see if we can capture any more. So we'll continue those efforts. But if you jump 2 slides, it's an example of the type of information that we've been capturing. So for each 2-year study, then for each compound, we've captured the strain, the sex of the animals that was used, the target organs where tumors were observed, the specific tumors, and then also the tumor incidences at each dose. And so there's a lot of information here, but it'll help us downstream to monitor, to model our transcriptional responses and the degree of activation of the MIEs and correlate that with the tumor incidence. Okay, and then last but not least, the gene signature development team finally in 2022 really was able to get started in earnest now that we have all the data compiled and many of the annotations coming in. We started with PPAR MIE because that's what we had the annotations on the soonest, but are now expanding to additional MIEs now that much of the annotation is complete. So our first goal then is to derive consensus gene sets which read out on the activity of each of the MIEs. And so various team members are taking a number of different approaches to this. For example, for PPAR and PXR, we've looked at t-test and random forest for deriving those signatures. And then more recently, the NIEHS has been applying some machine learning approaches, looking across now all the MIEs and developing signatures for each one. As was brought up earlier with the DDI discussion, we realized it's going to be important to align on a consistent approach here, to report all our methods, to really document this. So that's something that the team is working through now and aligning on which we think are the best approaches to lead to our consensus gene signatures downstream. I do want to point out and just kind of pause. We can go next slide a little bit to point out that what we've done so far is really just step 1 of identifying the gene sets. And so what we've done today is shown in that example A there, where for a particular MIE, we take the compounds that induce that mechanism, say AHR. So the list of compounds that induce AHR, we look at the downstream differentially expressed genes and ask what are the common genes activated by each of those compounds. And we're doing that to identify the most sensitive genes responding to that particular MIE. The limitation with that, though, is that almost all compounds induce multiple different downstream gene expression networks in the liver. And so just by chance, you'll often get overlap in these signatures that isn't necessarily a direct downstream target of your MIE of interest. So this helps us to identify sensitive signatures, but there is a possibility there may be genes that come through there that aren't specific for that MIE. So we're taking two subsequent approaches then that'll come next to help us ensure that the genes underneath each of our signatures are as specific as possible. So the one approach is there in B, and this is by looking at the totality of the data. So we can use approaches sort of like Jim Stevens' group has done to identify what are all the transcriptional regulatory networks, so all the co-expressed genes within the liver, and then ask the individual genes within our MIE signatures, are they specific to that biology, or do they also respond to other biology? And we've taken a similar approach at Merck with this multifactorial regression approach, where we specifically select genes in our signatures that are modulated by one MIE, but not any of our other signature sets. Then the other approach, Chris will talk about in depth in a minute, but that is the rat knockout studies we're doing. So these are knockout studies for each of the MIEs where we're treating with multiple different compounds. And so that lets us look if that specific MIE transcription factor is required for the transcriptional responses we're seeing. So we'll do that using RNA sequencing data, and it also provides the opportunity to do ChIP-seq analysis in order to confirm that our genes and our signatures are indeed direct downstream targets of the transcription factor of interest. And so putting all that together will help us derive the most sensitive, specific, and proximal signatures of each of our transcription factors of interest. So I labeled on that slide, you'll notice this said part 1, and because identifying the gene sets is only the first aim of the working group. And so you can go next slide. With those signatures in hand, we'll be able to develop thresholds of biological activation, or I called it POD here, but activation beyond what we see in a baseline for each MIE. But that biological activation isn't necessarily of carcinogenic concern. And so part 2 then becomes understanding how much do we have to activate each of these MIEs to have an association with carcinogenesis risk at the end of 2 years. And then step 3, we'll be looking across all the MIEs and seeing if there's additive effects. If we get a medium activation of MIE 1 and 2, does that lead to a concern with a different threshold than the individual MIEs? So I'll expand on that part 2 then in the next slide. And so we know that low or transient levels of activation of the MIEs is not necessarily a concern for tumorigenesis. And so an example there I'll give is omeprazole, which is a really strong AHR inducer, but it induces it within the first 24 hours, and then that induction rapidly declines over time. And at the end of 2 years, it's not associated with tumorigenesis. Compare that to something like TCDD or PCB126, which induces a similar level of activation in that 1 day time point, but the activation is maintained or even increased over time. And that's the profile of concern at the end of 2 years. And so we laid this out. This is a figure. The figure on the left there is from that 2022 Toxicology paper that the outreach team put together. So we lay that out in detail there. So how do we identify these thresholds of activation that will be associated with carcinogenesis? And that's where the annotation of the 2-year outcome data with all the doses and the tumor incidences becomes really important. So we'll need to be going through that for each MIE and deriving the activation level at each time point that's associated with tumorigenesis. And again, we do have proof of concept in the literature. Work from both Chris's lab and for Merck has shown that this is doable, and there are, levels of MIE induction that don't lead to tumors, but then above a certain threshold where you see that propensity to lead to tumors at 2 years. Next slide. Okay, so just last slide, leave you with our timeline. Then I've hopefully relayed the large amount of work the team has already accomplished since 2019. Just aligning on the approach, the MIEs, the analysis platforms, what data sets to pull in, getting all those data sets QC'd and ready for use. The huge amount of work going into compound annotation that is nearing completion. And then excitingly now starting to construct the biomarkers, really entering the fun stage of the project. And then I'm going to transition to Chris, who's going to talk about some of the new studies that we're performing, specifically the knockout rat studies for each of the MIEs.
All right, Chris, if you're there, you should be, yep, see you, should be able to talk now.
Very good, thanks. Yeah, hi everybody. So I'm going to go ahead and just briefly talk about the rat knockout study that we put together that's going to be useful for identifying the genes that are specifically linked to the activation of these different MIEs. So next slide, please. So Keith touched on this a bit, talking about some of the potential caveats about using only chemical induced data to generate gene lists that are associated with an MIE. So for instance, there could be off-target effects, there could be a major confounder. We do have examples where there are chemicals that activate more than one MIE, in particular those that activate both PPR alpha and CAR. It's also possible that there'll be genes that are identified using these different machine learning approaches that are in fact are being dragged along and regulated independent of the fact of being predicted. So using the existing data, which is all chemical induced profiles, we can't necessarily systematically identify the factor dependent genes without having more information. Now we could do this, in theory with, for instance, ChIP-seq data. But we thought this was an opportune time to propose a new study, which has been approved by HESI, and we're now it's now underway to use comparisons between wild-type rats and knockout rats to identify those genes which are regulated by the factor. Next slide, please. So, the argument for doing this study is that, go back one. So the argument is that by doing these analyses, and doing these comparisons, it allows an unequivocal assessment of factor dependency. And there are many examples of this in the literature. And we feel that the lists that we generate from this type of analysis could be used two ways. So they could be used to build the biomarkers de novo, and then to test them on microarray data that's annotated for factor activation, or to help to validate the chemical-centric methods that we're currently using. And so the next slide just shows the examples in the literature which help give us some confidence that this approach is going to work. And so I think we started building biomarkers for predicting mouse liver MIEs back in 2013. And we took advantage of comparisons that were already published in the literature, where groups had been comparing effects in wild type mice and in various null mice. And so the concept here is to look at a number of chemicals that are known activators of a particular factor in both wild type mice and in that factor null mouse, and then comparing those to your controls. Go ahead and click it one more time. Yeah. So, and this is just one way of doing this, but it seems to work in that we can generate these gene lists and we can filter them looking for genes which have certain characteristics, those of which are either upregulated or downregulated in the wild type mice, but not in at least in the same direction as the knockout mice. So for instance, with the PPR alpha example shown on the far left, we looked at the expression patterns of phenofibrate, PFOA, and YF in the wild type mice. And then we filtered out those genes which are also regulated in the same direction as in the PPR alpha knockouts. So we can then average across the comparisons in the wild type mice to generate this final gene list, which earlier we called a signature, and then now we've now call it a biomarker, and then use that biomarker to then compare to gene lists which are in various databases. Next slide. And so without going into too much more detail, what we found was that these mouse biomarkers have excellent predictive accuracy. We've made now 6 of these things, and they have predictive accuracies of 94% and above.
Sorry,
So
some of the slides are on a timer, Chris. So sorry about that.
Okay. Well, I better talk fast then. Okay. So you can go to the next slide. So this is the study plan for what we plan to do. And I'm sorry about that timer. I didn't realize that was on. Yeah, you can go ahead and just take it off.
Sorry, let me get out of the PowerPoint and then maybe
Yeah.
that'll be okay. I think this is what we did before. Sorry, everybody.
Yeah, that's
Okay, is that big enough to see?
it's fine for me.
Okay.
Yeah, it should be fine for the audience. Okay, so here's our plan that we have now settled on. And we are lucky to be able to recruit an excellent investigator, Dr. Udayan Apti from the Kansas University Medical Center, who is going to be conducting the exposures in his lab. He has extensive experience in carrying out rodent exposure studies. And so we spent a lot of time trying to figure out exactly what would be the best set of conditions for doing the exposures. And for instance, and also where would the knockout rats come from? So we're now planning to expose 6 different strains of knockout rats, and all of them, except for the estrogen receptor alpha knockouts, are coming from Inotiv. So we have to look for genotoxicity. We have the P53 nulls. For non-genotoxicity, we're going to be looking at AHR, CAR, ESR1, PPR alpha, and PXR nulls. So you may notice, well, we don't have any knockouts to specifically look at cytotoxicity, but we can probably fill that in later. What we decided on, and this is a balance between sort of best science and the realities of our budget, but we would focus on males only. And I will throw in the exception of the ESR1 knockouts, in which both males and females are going to be treated. And that's mainly because we've been doing everything in males thus far, but it's the females that actually get the increases in the liver tumors. You're not sure about what the outcome would be in male rats after exposure to some estrogen receptor activators. So for each strain, we plan to treat them with 3 reference chemical activators. And some of these have been determined by the Carcinogenomics work group. And we're going to have 1 time point of exposure. Now, in parentheses, we have 3 days, but we haven't settled on that. It may be 3 to 5 days. I think we still need to discuss that. We're going to expose the wild type rats to 2 doses of each chemical, a low and a high, choosing those doses which are one being high as being a tumorigenic dose and one being subtumorigenic. And then we're going to look at expose the knockouts to 1 high dose. There'll be 5 rats in each block. The liver gene expression will be examined by RNA-seq, and Merck has graciously said that they'll go ahead and analyze all of the livers that we give them. We also have some opportunities for doing some additional studies, and there's some interest in doing some ChIP-seq. So my last slide. So basically what we plan to do is to identify these factor-dependent genes by doing these comparisons between the wild types and knockouts. And we can do this a number of different ways by integrating these different computational approaches to get 6 non-overlapping gene sets, which again can be used either to build predictive biomarkers or to validate the ones using the chemical-centric approaches. And the exposures are expected to start in 2023 in blocks of studies as the rats become available. Okay, I think that's it. Thanks for listening.
Great, thanks, Chris. Thanks, Keith. Folks can, if they have any questions, go ahead and put them in the chat or raise hands. You know, and just hearing the conversations today, I think the really important piece, and going back to what was mentioned with TGX DDI, will be the recording, so that, you know, with the ultimate goal, if we can get these qualified, we would have all those documentations. I actually see Carol with her hand up now. So go ahead, Carol.
Yeah, thank you. This was a fantastic summary. Congratulations to the team on the incredible progress in the past year. It's wonderful to see this coming together. I was curious about what's in the AOP wiki as it relates to this project, because you know, as we're building towards regulatory acceptance, thinking about the OECD, having AOPs in there as well for the MIEs leading to the various cancers of interest would be very useful for demonstrating application in IATA. And given the extensive amount of work that the students have done in collecting all that information about the MIEs and then the information that you have on cancer, and Chris, now you're generating the essentiality data with the knockout studies, you've pretty much got everything you need for AOP building. So those endorsed AOPs could really help you out in the long run.
Yeah, I mean, I agree with you, Carol. I propose to put together the PPAR alpha mode of action many years ago. And you know, you can find it in draft form, for instance. But the hard part is that it's, I guess, it's similar to your validation project, yet the bar just keeps changing, and that there's new requirements, and in a way, my group has been waiting for things to settle down, where we have strict rules as to what information they need and how to convey it.
In the AOPs, you mean, in AOP development?
Yeah, I mean.
Yeah. Okay.
Right. So and I'm pretty sure that all of the MIEs that we're working with are linked to the liver cancer AOPs are already in the AOP wiki. It's just that some of them are, I think probably most of them are still in draft form.
Carol, did that answer everything?
Sorry, I got muted there again. Yeah, that's. I mean, I'm not surprised, Chris. It's a high bar to get those AOPs endorsed, but I was wondering if maybe the students, who have been doing this work, could take that a little bit closer to the finish line because they've got extensive amounts of data there, and it is pretty settled now. I can help, I guess, in some coaching to help get it to the finish line, if you think it's important and you'd like to try.
Yeah, well, thanks for that offer. I'll consider that. That might be one way of getting us out of this dilemma, because I can never seem to find enough time to finish Peter Alpha.
Yeah, thanks, Carol. I mean, it's information we've collected that's from the public domain. If that's helpful to share too, I'm happy to facilitate, or if we can come up with a way that's maybe not too onerous to get that to the folks developing those AOPs, not have it all fall on Chris. Great. Udine, I see your hand up. You should be able to unmute now.
Yeah, hi. I think my picture's coming up, but this is a great talk, and I'm really proud to be part of this study. And we are just wanted to report that we have already begun with the ESR1 part of it, and hopefully get going with the others. I had a question though, and it's more of a philosophical question, I guess. And that is, I've done a lot of work with human liver cancer, working with patient samples and molecular mechanisms and things like that. And when we think about the mechanisms that drive that, a lot of work has gone in, there has been done by people like Jessica Zuckman-Rossi and Tanya Raskams and Paul Monga to identify what we call in toxicology AOPs. They don't call them AOPs. These are all liver doctors or pathologists, human pathologists. They include, majority of the landscape there is associated with mutations of beta-catenin, Wnt signaling. The only AOP that is sort of common between what we see in the human world there and what we have is P53. So I'm just coming from a sort of a different angle there. I was just wondering, the goal here is really to stay focused with the rat studies, I think, right? I mean, we are not that interested in mimicking what happens in human population, because this is focused at figuring out what goes on in our animal studies, because that's what we're trying to find an alternative for. Am I correct on this analysis?
Yeah, I think you are. So, in a way, some of the work that we're doing, we already know that these AOPs are not relevant to humans, right? And that's part of the point. We want to provide an opportunity to use these tools to identify chemicals that would cause liver tumors that are irrelevant, and then we could, then that group could argue for a waiver. Right. But the landscape of the regulatory arena, though it's moving towards, in vitro and silico, still relies on, an understanding of the molecular basis for tumors before making a judgment. Right. And sometimes those regulatory agencies, they change their minds about the human relevance of a particular mode of action. And so it's sort of, we can provide the tools, we can provide the biomarkers, we can provide the threshold levels to be able to say, or any group to say, yeah, this chemical would cause a liver tumor in a 2-year bioassay, and it would be through a CAR mode of action. Well, some regulatory agencies are going to handle it, one way, and maybe in Europe it might be handled another way. but so I think we're going to stay out of that arena. What we're doing is already, time consuming enough to provide those tools for then interactions to happen between, a sponsor and a regulatory agency.
That's great. I have two other comments, and one is that in the same aspect, once we finish these studies, we'll have samples and we can look at human relevant AOPs as well, because we've done this. So for example, if our chemicals activate AHR or CAR or PXR, and whatever we're doing in those rats, we can see a concomitant activation of say beta-catenin or Wnt, and that's very easily doable because we'll have the sample. So that's not a problem. We can cover that. And the other thing I wanted to mention, this is really a comment for the whole group and the audience, is that when we do these studies, we have a plan to take the liver, and we have planned everything we want to do it. But these, there are other organs in these animals that may be of value. And so if somebody is more specifically interested in something, spleen, lungs, kidneys, whatever, I would like them to reach out to us as soon as possible so we can sort of plan that collection in the process, and they can do whatever they want with those tissues. I'm a very hepatocentric person. I always tell my students, when I see a rat or a mouse, I'm looking at a big liver with a tail. But there might be others who are interested in other organs. So they are welcome to take the tissues as long as we coordinate up front, and I'll be happy to help.
Great, thanks, Udayan. Question from Carol came in, and we've talked about this some on the team, but it would be nice to discuss here. Have we found sufficient data for each MIE, clarifying a large enough training set using the existing data, or will there be enough left over for external testing? So the way I see this is kind of like our small n issue that we've come across, that for some of the MIEs, we really only do have a couple compounds. For example, estrogen, I think we had 6. Maybe Keith, I know you've addressed this before. Would you like to maybe start to take that on?
Yeah, no, it's a really important question. So thanks for asking. I think for identifying the gene sets, we're in pretty good shape in that for most of the MIDs, we have quite a few compounds. It becomes more challenging in identifying the thresholds for carcinogenesis, just because of the limitation of what's been run through 2-year carcino studies. So for example, for AHR, I think there's really only 2 compounds where we have nice dose response data in the 2-year carcino study to benchmark against. And so it's definitely be something that we need to tackle. I don't necessarily know the answer up front. As far as the individual gene signatures, I'm more confident there. And then using some of these other approaches of, again, we're not working in the dark. Most of these MIEs have very well-known canonical gene sets, right? We have ChIP-seq data showing direct binding to promoters. So there's a lot of sort of weight of evidence that goes into those gene sets, which helps us build confidence, even though we have a somewhat small compound set. So from a purely machine learning standpoint, you do have a small n problem. But since we're working with known biology, we can leverage all that literature to support our conclusions as well.
Thanks, Keith. And Chris, I don't know if you have anything to add to that.
So this is it's a potential problem. I'm not too worried about it based on what Keith said. And I did write something, a request. If anybody does have any studies where they want to share data or even possibly livers from rats treated with chemicals or maybe reference chemicals, the CAR-C outcome in the liver, that would be quite useful. We're particularly interested in RNA-seq data derived from the livers of rats treated with subtumorigenic doses of chemicals. As Keith said, that we would need that to help derive the threshold for tumorigenicity. But, one thing I will say as an aside, with Scott and others, we have a paper in going through our internal review process, which indicated that, based on one of Scott's data sets where they were comparing RNA-seq and AFI data, that it looks like the thresholds that we developed using AFI work just fine with RNA-seq data. And that's because I think you're at a low enough level of activation that you can, it doesn't really change the, ability to pick up the tumorigenic doses. So, but we need to return to that and determine that experimentally. But more data, is always better in my mind.
Thanks, Chris. I don't see too many more. If folks have questions, they can raise hand. I was mulling on something. We're going to talk about error-corrected sequencing in depth tomorrow. Are there thoughts on, say we go through these exercises, we create our biomarkers for the transcriptomic side. How would you maybe pair the error-corrected? And specifically, what if there was conflicting results, like you saw activations of certain pathways in the MIEs, but a lack of response in the error-corrected? What's your thoughts there? Or is it too early to know?
I think it's too early to know, right? I think something we'll see is the flip, right? And one of the advantages of the error-corrected sequencing approach is it's not tied necessarily to upstream mechanism, right? So we're looking at a set of 6 mechanisms, but there's alternative potential ways you could pick that up by the error-corrected sequencing, right? So that's something you could miss, potentially have a false negative by gene signatures that you'd pick up, and then vice versa. But yeah, when we have, I think we still need to do those studies, what's the sensitivity of the error-corrected sequencing for each of these MIEs, still TBD.
Thanks, Keith. Anything from you to add to that, Chris? Oh, you're muted. Asked to unmute. Maybe just no. Oh, there you go.
It is no.
Okay. Well, good. We don't see too much else in the chat. I really appreciate this overview. I think as Chris called out, the additional samples would be really helpful. I think on the bioinformatics side, we have a really great group of people helping us, but that is somewhere we're open to more support. So if folks are interested in being a part of this initiative, please feel free to reach out. And thanks to Keith and Chris for all their leadership, and all the other leads like Hydrin and Udayan and Pierre and Alexi have done a ton of work for the team. So thank you all.